This is a lecture from October 23rd, 2020. And in this lecture, we're going to discuss how skeletal muscles change with age. And so in the previous lecture, we spent time discussing how skeletal muscles function. And in this lecture, we're going to kind of discuss how that function might decline and what might contribute to the decline in function of skeletal muscles over time. And we're going to first, in this part of the lecture, discuss the correlation between strength and muscle area and how that changes with age. And then we're going to discuss a little bit about how the innervation of muscles changes during aging and how that relates back to aging muscle atrophy. Right, and so we all have sort of seen anecdotally that muscle mass is lost over time, right? And this non-pathological condition where muscle mass is lost in a time-dependent manner is known as aging muscle atrophy. And atrophy is just a decrease in size. And so this is the decrease in size of muscles as you age. And as I said, this is non-pathological. So this is not related to disease in any way. This is just kind of a natural progression of aging that results in this decreased muscle mass, right? And so the results of decreasing both muscle cell size as well as muscle cell number is what actually causes aging muscle atrophy. And you can see here, this is a cross-section of two quadriceps of a 40-year-old triathlete. And this is an MRI image, right? And so you can see the muscles here and adipose tissue on the outside of both legs. And then with aging, what you can notice is that this muscle tissue greatly decreases, right? And it's very obvious even to somebody who's not trained at looking at MRIs. And what you'll notice too is that there's this more accumulation of sort of white tissue. This is the adipose or fat tissue. And this individual was sedentary, and sedentary doesn't mean like bedridden or totally unable to move. This is just an individual who moves kind of at a normal rate. So they maintain their body posture, they walk a little bit, enough to kind of get by. But this is not somebody who has totally stopped using their muscles altogether. It's just somebody who does not add any additional physical activity into their routine, right? And so over time, quadriceps volume in elderly versus young individuals, so age over 70 versus under, quadriceps volume decreases, and muscle mass can decrease at a rate of about 8% per decade for men and 6% per decade for women. What's interesting about this loss of muscle mass or aging muscle atrophy is that it can actually be modified by physical activity. And so you'll see here a 74-year-old sedentary man's quadriceps, but a 70-year-old triathlete's quadriceps in this image here look almost identical to this 40-year-old, right? And so if that doesn't make you want to go out for a run, I'm not really sure what will, right? And so for individuals who exercise, they can actually attenuate this loss of muscle mass or this aging muscle atrophy and reduce it from 8 or 6% per decade to only about 2%. And as a kind of overall result, aging muscle atrophy can be delayed about a whole decade of your life. And so in the case of most of the college students listening to this, that would be about half of your life. It's about a third of mine, right? And so you can delay this loss of muscle mass just by increasing your physical activity and using these muscles. And so in addition to just kind of a loss of tissue, there's also a loss in both strength and power generated by skeletal muscles that's associated with age. And so muscle strength and muscle power are highly correlated with the cross-sectional area of a muscle fiber, right? Or basically the width of a muscle fiber. And the higher the cross-sectional area, the more power and strength a muscle cell can generate. And as we saw in the previous images, cross-sectional area decreases in elderly individuals. And you can see that in this graph here as well. And this is true of both the type 1 or the slow twitch fibers, as well as the type 2A or the faster twitch fibers. In comparison to young individuals in the white bars, the elderly individuals have a decreased cross-sectional area in both of these different fibers. And that loss of cross-sectional area correlates highly with a loss of strength and power at the same rate. Right? And down here is a measure of just kind of force or power generated by these particular types of fibers, type 1 on the left and type 2A on the right. And what you'll note is that in both type 1 and type 2A, in elderly individuals, there is a decrease in the amount of power generated per unit of area of the muscle cells. And it's important to remember that a decrease in cross-sectional area reflects not only the decrease in width of muscle cells, but just a decline in the number of them as well. Because declining numbers of cells will decrease the width of the muscle overall. And type 2 fibers are generally lost at a higher rate than type 1 fibers in older individuals. It's important to note that they both decrease, right? So you do lose both types of fibers, but you lose more type 2. And this actually makes sense when you think about the function of type 2 muscles. Muscle fibers is to be more force generating, more strength generating, more for quick, fast movements. And what we see in terms of motility of older individuals, they move slower, they lose their ability to respond quickly to those kind of things, as well as to generate the force and power. And this corresponding decrease in the amount of type 2 fibers helps explain why we see those phenotypes. And in terms of the mechanisms that actually underlie this muscle atrophy, there haven't really been a lot of causal mechanisms identified. There is some changes in terms of mTOR as well as FOXO signaling that change the metabolism in aging muscle. But this is not muscle specific, right? This is kind of general among all tissues in the body. And so why it would affect muscle aging more than any other tissue is not really understood. One other hypothesis is that a loss of mitochondrial function that occurs with aging could increase reactive oxygen species within the muscle, right? And this is a relatively good idea to think about, right? Because as we said, muscle cells have a lot of mitochondria. They require a lot of energy. And so as mitochondrial function declines, there's a lot more mitochondria that could be generating a lot more reactive oxygen species and damaging the biomolecules within the muscle cells. And indeed, there is an increase in oxidation of myosin fibers in aged muscle. And so that is something that has been observed, but it hasn't been studied well enough to be identified as a specific cause of the muscle atrophy seen in older individuals. But one thing that we do know for sure declines with age and can contribute, or is at least correlated with a decrease in muscle mass that we see in aging muscle atrophy, is the decrease in motor neuron innervation of muscles, right? And so with age, motor neuron axons can actually thin, they elongate, and that can ultimately lead to their degeneration and potentially motor neuron apoptosis or cell death. And you can see here a motor neuron process in green, right? These are the axons labeled in green. And then acetylcholine receptors labeled in red, right? And acetylcholine receptors are on the surface of the muscle. So you can see basically the neuromuscular junction in these pictures where the neuron, this motor neuron in green, is meeting the muscle in red. And what you'll notice as in these images, comparing a mouse that is 3 months old to 2 years old, the motor neuron axons, or these processes, do elongate. They extend out. And as a result, it takes a lot longer for an electrical impulse to reach the NMJ, or the neuromuscular junction. And it can actually degrade the signal along the way, right? And so that can decrease stimulation of the muscles, and then can lead to some of their atrophy as well. And over time, there's about, between a 20 and 30% reduction in motor neuron innervation observed in older individuals. And this occurs more in the type 2 fibers. And once again, this can kind of explain some of the phenotypes we see, right? Where individuals who are older can no longer generate force and power with the same amount of force and power as a younger individual. And that could be due to their decreased motor neuron innervation of type 2 fibers, right? And so there's also this idea of, if you don't really use it, you lose it with muscle. And we discussed that in terms of cross-sectional area, right? So that sedentary individual from the beginning of the lecture loses their muscle mass as a result of not engaging it via physical activity. And the same is true for when motor neuron innervation declines, right? Because you're not stimulating the muscle, you're not promoting it to move, you're not activating it via innervation, and therefore you kind of lose it. And so when the neuronal processes lengthen at the NMJ, the whole neuromuscular junction can actually end up undergoing fragmentation. And what that looks like can be seen up here in this image. And what happens on the neuron side is easy to see, I think, the extension of these processes. But what happens on the muscle side is that acetylcholine receptors that actually, respond to the neuronal signal on the muscle side of the NMJ are organized into these things called folds. And so what you can see here is basically acetylcholine receptors in white outlined. And they look like this pattern because they are on the surface. And then there's kind of an indentation in black. And then there's another fold up where there's acetylcholine receptors, and then an indentation down. Another fold up where there's a lot of acetylcholine receptors, and another indentation down, right? And so these folds give acetylcholine receptors sort of the best possible access to the neuronal processes, right? By keeping them on the top of the muscle cells, right? And not in these indentations down here. But what happens over time is that the folds can actually fuse together. And when they do that, acetylcholine receptors are no longer exposed to the neuronal processes. They can end up in the indentations rather than on the top surface. And that fusion and the decrease in size of these folds means less acetylcholine receptors, which means less responsiveness to neurons. And without that ability to respond, sometimes you'll lose it, right? And the NMJ will undergo fragmentation because there's no longer innervation, and there's no longer a way for the muscle to respond to that neuron in the particular location, right? And so with age, we do see this decline in motor neuron innervation, and that likely contributes to the aging muscle atrophy.